Welcome back, everybody. So today what I want to do is talk to you about mitosis, that process by which the DNA is separated when two new daughter cells are forming. And before we get started, let's take into account some things we want to think about. The goal of mitosis is to separate the sister chromatids and then to form two new cells. And so in humans, each daughter cell must get 23 pairs of chromosomes. So that's a lot of individual chromosomes to separate. We're going to distinguish at times today mitosis, which is the actual division of the replicated DNA in the nucleus, from cytokinesis, which is the division of the cytoplasm and the separation of the two cells. You should understand that mitosis is a continuous process and it can be divided into a series of stages. And each one of those stages is defined by a particular event in the process. Conventionally, it's divided into 4 or 5 phases, and they have names like Prophase, Metaphase, Metaphase, Anaphase, and Telophase. That's the 5-stage model. When it's only presented as 4 stages, Metaphase goes away, and we refer to late Prophase. And in a little bit, you're going to learn what each of those phases means. What are the criteria we use to say that a cell is in a certain phase? And cytokinesis, that process of separating the two cells, is usually well underway by telophase. Now, before we go on, I want to talk about some confusion that arises. The word mitosis is used in 3 different ways. One, it is used often just to refer to eukaryotic cell division. So people will talk about cell multiplication by mitosis. And that's not referring specifically to the separation of the DNA, but to cell division itself. 2, if we look at the cell cycle that we saw earlier, we talk about the mitotic phase, which contrasts with interphase. And that's the phase during which both mitosis and cytokinesis happen. So the mitotic phase is that phase in the cell cycle where mitosis, the separation of DNA itself, of the sister chromatids, and that's the 3rd definition of mitosis, and cytokinesis, the separation of cells, occurs. Here is a slide that just lists the key events in the process of mitosis. The first is the condensation of the chromosomes, and what that means is the chromosomes actually become thicker and more dense, and we can actually see them if we stay in the cell. Second is the formation of the cytoskeleton apparatus that's necessary to move the chromosomes from the middle to each side of the cell. The cytoskeleton apparatus consists of the centrosomes, the centrioles, what's called the spindle and the aster, and we'll make those clear in a little bit. Third is the migration of the centrosomes and cytoskeleton apparatus to each pole of the cell. Fourth is the dissolution, which means the dissolving of the nuclear membrane. Fifth is the attachment of chromosomes to the cytoskeleton spindle, and we'll see pictures of that in a second. Sixth is the alignment of all the chromosomes on the equator, on the equatorial plane of the cell. Seventh is the separation of the sister chromatids to opposite sides of the cell. Eighth is now the reformation of the nuclei in the daughter cells, and then the decondensation of the chromosomes. In other words, the chromosomes relax again, and you can no longer see them as individual chromosomes. And then finally, 9th is cytokinesis. So we're going to go over this several times today so you really get an understanding of this. Here is a picture of it. And at first, this looks really crazy, okay? I'm going to go over it very briefly. Here is prophase, the first phase, and notice what's happening. The chromosomes are condensing and the spindle is splitting and starting to move to the poles. In metaphase, now the nuclear membrane is completely gone and the cytoskeleton has moved to the poles, and we'll talk more about this, and I'll give you time to write down the definitions of this later on. In metaphase, the chromosomes, the replicated chromosomes, all line up on the equatorial plane. And since cells are spherical, they have a whole circular plane on which to line up. Then in anaphase, the sister chromatids are pulled apart. In telophase, the new nuclei begin to reform, the nuclear membrane reforms. And notice that cytokinesis is already happening. The cells are beginning to separate. And then that's what happens finally in the very last stage is cytokinesis, the separation. You can study this later. We're going to move on for now. Within a cell, mitosis is a continuous process. When each phase is reached, the cell doesn't stop or rest, but rather the process just continues. Thus, the 5 phases are simply words we use to describe this continuous process. It will be important for you to learn the defining characters of each phase so you can identify the phases that you will observe. And once you think about this whole process, there's a certain logic to it that will make learning these phases rather easy. So let's look at them now. So in prophase, the chromosomes begin to condense. In other words, instead of being spread out all over the nucleus, they become more compact, and they therefore become visible as actually linear chromosomes. In addition, the nucleolus disappears, but the nuclear membrane is still intact, and the centrioles begin moving, or the centrosomes, I should say, begin moving to the opposite poles, and the spindle, which is the thread-like fibers that arise from the centrioles, and I'll show you that in a picture in a second, begins to form. That's prophase. In pro-metaphase, the chromosomes are fully condensed. The nuclear envelope now disappears, and the chromosomes no longer appear contained. The spindle starts to extend towards the middle of the cell because by this point, the cytoskeleton has moved to the opposite poles. And we'll talk about the aster in a second. And the chromatids begin to attach to a microtubule of the spindle. That you cannot see, but that's happening as well. So here are pictures. Here's interphase, and look, you can see no chromosomes. But now in prophase, at the first point, you can see that the chromosomes have condensed, but you can see that the nuclear membrane is still intact. And here's the spindle apparatus that we're talking about. And here they go. These 2 replicated centrosomes are moving to the poles. They're moving this way. Now by pro-metaphase, look what's happened. The chromosomes are fully condensed, and you can see them individually. The chromosomes are no longer contained inside a nuclear membrane because the membrane is gone. And the spindle apparatus has moved to opposite sides of the cell. So that's prophase and pro-metaphase. Let's take a look at the next 2 phases, which are the easiest to identify and remember. In metaphase, the chromosomes line up on the metaphase plate, which is the equatorial plane, the plane at the equator. And the cell waits until all the chromatids are attached to the spindle and lined up on the equatorial plane. It has some sort of checking mechanism before the chromatids will separate. In anaphase now, the sister chromatids begin to pull apart. And anaphase ends when the daughter nuclei begin to reform and the chromosomes begin to decondense. Let's take a look at these in pictures. So here's metaphase, and this is hard to see, but what's going on here is all the chromosomes and their centromeres are lined up here, which is the equatorial plane. Here are the 2 spindle apparatuses, so you can see this is one pole and this is the other. And now in anaphase, look at this. The sister chromatids are separating and being pulled apart. They're moving. This group is moving down and to the right. This group is moving up and to the left. That's anaphase and metaphase. Let's finally look at telophase. In telophase, the nuclear envelope in the daughter cells reform. The chromosomes completely decondense, so they are no longer visible. If you stained at this point, you would not see individual chromosomes, just the stain spread over the whole nucleus. And when the 2 nuclei form, mitosis is over. Cytokinesis, the cytoplasm is cut. And as I said earlier, it's almost always underway by telophase. So let's look at this in a picture. And here it is. Here you can see now that the chromosomes are decondensing. The nuclear membrane is reforming. And so we are now done with mitosis. And you can see the cell is beginning to split apart. That's cytokinesis. Here now is a picture of mitosis in one slide. Interphase, the chromatin is spread all over the place. Prophase, the chromosomes condense, and the spindle apparatus, the centrosomes, begin to move to the opposite poles. By metaphase, the chromosomes, the individually replicated chromosomes, are lined up on the equatorial plane. In anaphase, the sister chromatids are being pulled apart. In telophase, the nuclear membrane reforms and the chromosomes decondense, and then cytokinesis is the final separation. You will note that this diagram shows the 4-phase model. It doesn't distinguish between prophase and pro-metaphase, but you'll have to learn that. Next, what I want to do is just talk a little about the centrosome. It is the structure that forms the spindle and contains the centriole, and it moves to each pole of the cell. The so-called aster are cytoskeleton elements that reach from the centrosome outwards to the cell membrane, and they anchor the centrosome and the whole spindle apparatus on the cell membrane, and we'll see a picture of that in a second. Microtubules radiate from each centrosome towards the center, towards the equatorial plane, and form what is called the spindle. Some of these spindle elements attach to a complex of proteins that's associated with the centromere, and they're called the kinetochores. It's in parentheses because that's not a vocabulary word you need to know in this course. And then motor proteins, which we saw earlier in vesicle transport, are used to walk the sister chromatids along the spindle to the incipient cell towards the poles. So we'll see a picture of that too. So here is metaphase. Here is the spindle apparatus right here. This is the spindle. And look, going upwards in this, out towards the cell membrane, is the aster. So here is the aster. So once again, this is the spindle and this is the aster. And right here in the middle is the centrosome. And here is a schematic diagram of it. You can see there's the aster, which is attaching to the cell membrane. Here is the spindle. Some of those spindle elements are attaching to the centromere, and they're going to allow a motor protein to walk the chromatid towards the new cell. Here's a picture of the motor protein walking along a microtubule in the spindle, and it's carrying the chromatid to its new nucleus. Finally, I want to look at cytokinesis. In animal cells, cytokinesis occurs by a process known as cleavage. The daughter cells separate and form what's called a cleavage furrow. Cleavage literally means to cleave something or to cut it in half. In contrast, in a plant cell, a cell plate forms during cytokinesis. So let's see what these look like. So here's an actual micrograph of cytokinesis in an animal cell. And you can see that this furrow is being pinched off, and we draw it schematically like this, and eventually they will separate. And what it appears to be is there's microfilaments, which are a type of cytoskeleton element, which just keeps, like a lasso, tightening, and drawing the two cells apart. In contrast, in a plant cell, we see the formation of cell wall elements and cell membrane elements. And so what happens is here is the cell plate forming between the two cells, and eventually it will close and form a new cell wall. We're not going to get much into how plants do that and how those cells grow after this. We'll leave that for a more advanced biology course. And here are some pictures of the 5 phases in a plant cell. So here you can see this is prophase. How do you know that? Because the chromosomes are beginning to condense, but the nuclear membrane is still there. This is pro-metaphase because the chromosomes are fully condensed. It doesn't yet look like the nuclear membrane is broken down completely, but it probably is broken down a little bit. Metaphase now, the replicated chromosomes are along the equatorial plane. Anaphase, the two chromatids separate and are being pulled to the pole. This is very late anaphase here. And then finally in telophase, you can see that the nuclear membranes are reforming, the chromosomes are decondensing. And look at that, there's the cell plate forming. So that's it for today. What you really need to get out of this are the 5 phases. You need to remember what they are, you need to remember their order, and you need to know the criteria by which each phase is determined. And we're going to reinforce this in a lab where you're going to look at cells and determine what phase of mitosis they're in. So, until next time, go outside, look at nature, ask biological questions, bring them back to class, and I'll see you soon.